I'm honored to welcome you once again to our second of our fall series of our virtual lecture series. Today, we're really pleased to offer this very fascinating conversation with our colleague, Kamena Kostova. Dr. Kostova is a cellular biologist who studies ribosomes. Ribosomes are these tiny factory-like structures that inside cells that decode our genetic information and then use that information to produce proteins. And of course, this means ribosomes are absolutely critical for life. And when they malfunction, that can result in a wide range of very serious diseases like neurodegeneration and like cancer. Dr. Kostova investigates these fundamental questions of how cells respond when their ribosomes malfunction. Her research relies on a broad range of functional genomics methods, which include CRISPR genome editing. And you may have heard last week, Jennifer Doudna and Emmanuel Charpentier just won the 2020 Nobel Prize in Chemistry for their discovery of CRISPR. We've had the privilege, of course, in the recent past, of hearing both Doudna and Charpentier speak to us at our Capital Science Evenings here at Carnegie. Dr. Kostova and her lab were very early adopters of this tool, this remarkable tool, CRISPR, to study in her own research program. So we're very excited to hear what she has to say today. Dr. Kostova has been with Carnegie since November 2018, and she joined our Department of Embryology as a staff associate. And she came to us from the University of California, San Francisco, where she earned her PhD in biomedical sciences. While at UCSF, she was honored with the Fred Hutchinson Cancer Research Center Harold W. Weintraub Graduate Student Award. And before that, she received her BS in biology from the Massachusetts Institute of Technology. So like you, I'm very much looking forward to hearing what this exceptional scientist has to say today. So please join me in welcoming Kamena Kostova. Kamena?
Thank you, Eric, for this lovely introduction. And let me just take a second to share my screen with you, with all of you. Here it is. So thank you so much for the lovely introduction, and thank you all for attending this virtual seminar. I'm really excited to share with you some of the research that my lab has been doing. And I'm going to start by saying that I can't progress through my presentation. Wait a second. Let's see. Okay, here we go. Apologies for the little technical glitch. But I'm going to start by saying that we all live in a DNA world. And DNA is probably the most famous biomolecule. It is our blueprint, and it tells how our bodies are going to be built. But it also tells us if we're going to be predisposed to certain diseases, and holds a lot of therapeutic potential for drug design and personalized medicine. And you can tell how famous a molecule is by how many times it appeared on the cover of Times magazine. So, you know, DNA is kind of like the molecule of life, and it gets a lot of credit for that. But our bodies are actually not built from DNA. They're built from proteins. And these proteins are made by this very special molecular machine called the ribosome. So the ribosome is absolutely essential. It does a lot of work by building the proteins that allows your bodies to be built, but it gets very little credit for that. So if you want to take a one message from this talk, if you want to take a one conclusion, is that the ribosomes deserve a lot of credit for the work they're doing for us, but they're kind of the unsung heroes of our cells and our bodies, and they almost never get to be on the cover of Time magazine. So I'm here to do justice and tell you about this absolutely fascinating molecular machine. And as I said, the ribosome is the molecular machine that synthesizes proteins. The way this works is when the ribosome engages a molecule called the mRNA that carries a little piece of information that is copied from the DNA and carried to the cytoplasm. The ribosome hops onto the mRNA, it decodes the genetic message, synthesizes a protein, and then it's being released and can engage other mRNAs. So before you all log off from Zoom and go watch something more entertaining like the Great British Baking Show, I'm going to say that for this talk, we're not going to dig into the nitty-gritty details of translation and how this like RNA protein interaction happens, but we're going to kind of like take the bird's eye view of the process. And for the purpose of my talk, I want you to think of the ribosome as this little car that drives down the highway. And this is a metaphor that is especially appropriate for the type of research that I'm doing. So this process of translation is again very similar to cars that drive down a highway. Here you have this little ribosome that gets on the mRNA, which is the highway, by an on-ramp. It drives down the highway until it reaches a mark that's called the stop codon, and then it disengages, releasing the synthesized protein, and then the car can go and drive on other highways. So this is the basic process of protein production. And we know quite a bit about how these molecular machines are built, how the ribosomes are built, and how they get on the highway, how they drive down the highway, and how they're being released. However, what is very poorly understood in the translation field is what happens when mistakes start being made. What happens when you fail to assemble a functional ribosome, or what happens when this car gets into an accident while driving down the highway. And this is the major research focus of my lab. So we look at defects during biogenesis, as well as mistakes during normal translation. And I'm going to give you a little overview of both of these processes. So we're going to start with biogenesis. And building a ribosome is very similar to building a car. There are a lot of components that need to come together in a very highly coordinated manner. And we often think about ribosome biogenesis as an assembly line. And this is a picture of the Henry Ford assembly line. And here you see all these workers that work in a very coordinated, both spatially and temporal manner. And because this process is so complex and there's so many steps that are being involved, mistakes can happen. And when mistakes happen, this could be very bad for the cell. So for example, you can shut down the entire assembly line. For example, if this guy here gets sick and needs to take a sick day, then and there's no one to replace them, then the guys that precede him have no one to pass their partially assembled wheel to, and then the guys that follow him don't have anything to work on, right? They're going to run out of this like pre-assembled wheels too. So then everyone in this assembly line needs to take a sick day, and that's bad because you can't just have a day where you don't make any proteins in your body. In addition, if you make mistakes, if another worker, for example, is not very careful, so he every once in a while makes a wheel that's not assembled properly, and this wheel is not being detected, but it goes through the entire assembly line and gets attached to a car, this can also be very bad because you end up having cars that can get into accidents or break down or can have very deleterious effects. In the same way, if a cell does not assemble a ribosome properly, these ribosomes are released into the cytoplasm and they can malfunction in various ways. They can fail to synthesize proteins, or they can synthesize proteins by making a lot of mistakes, which can have very bad consequences for the cell health. So mistakes can be made, and we're very interested in how the cells ensures that you start with that you build a car properly, right? How do you start with the body, and then you add the wheels, and then you end up with a car that can drive? So we biologists like studying things by breaking them. So in my lab, we take this beautiful process of ribosome assembly and break it in as many possible ways as many ways as possible. So we engineer cells that, for example, make a lot of wheels, and there aren't enough cars for these wheels to be attached to. We make cells that build up cars that are missing a wheel, or the wheels are attached at the wrong location. And then for each of these mistakes, we ask, how are the cells dealing with them, right? Sometimes you have this excess components that, for example, you might store and use later, or here you have a mistake that is very easily repairable by just adding a wheel here. But sometimes you end up with a defective ribosome that is very hard to repair, right? That this is kind of like a terminal error, and you really want to send that one to the trash can and degrade it. So by making all these different mistakes during biogenesis, we're asking how are the cells dealing with the mistakes, and also what are the factors that say, oh, these are extra wheels, or oh, this car is missing a wheel, let's just put an extra one. And this is where we do high throughput genetic approaches such as CRISPR screening to identify the factors that facilitate these steps. And we have some very interesting ones that we're working on, and hopefully you can read about them very soon. So, right, some very big portion of my lab studies the quality control that monitors ribosome assembly. But once a ribosome is assembled, that's not the end of the story. In the same way, if you buy a car from a dealership, you can't drive it forever, right? Sooner or later, you're going to get a flat tire, you need to get your engine checked, or, unfortunately, sometimes accident happens. Same with the ribosomes. Even when they're perfectly assembled, once they start driving these molecular highways, there could be accidents. And similar to our human highways, the molecular highways don't contain a single ribosomes per mRNA, but there are multiple ribosomes that engage a message and translate it at the same time. So sometimes a ribosome can stop translating before it reaches the off-ramp and before it's released from the message. And then any other car that follows this stalled one is also going to get into this traffic jam. So why are these cars breaking down? Why are these ribosomes failing? Again, very similar to human cars, there are reasons such as low energy, like if you run out of fuel and the car cannot continue. For example, if the cells are being starved and they're missing key components to build proteins such as amino acids or tRNAs or many other factors. You can have highways that are kind of bumpy and have a lot of potholes. In the same way as the mRNA can have damage or mutations, and this car now cannot go through this defect here and reach the off-ramp. So again, it gets stuck. And last but not least, as we said, there could be defects on the ribosome itself. In this case, this car got a flat tire and cannot reach the off-ramp. So again, because these molecular highways are kind of two-dimensional, the cars that come behind the one that's stalled cannot just jump over it or go around it, but inevitably are also going to get stuck, leading to these molecular collisions where ribosomes start bumping into one another. And this could turn into a huge traffic jam because you can have more ribosomes coming down the highway, reaching this spot where you have the accident. So what we asked in my lab is, how are cells dealing with this problem? Do you have a lot of accidents happening that get resolved, or is the cell capable of preventing these traffic jams? And what we found is that the cells are actually very, very smart. And as soon as they detect these stalled ribosomes, as soon as they detect this first collision event, there are two factors that are called GGF2 and 4-HB, and they close the on-ramp. So when this on-ramp is being closed, there are no more cars that are capable of hopping onto the mRNA, and that decreases the stalling burden in the cell. So there are no more cars that drive to this place that has an accident until this accident is being resolved. So this is kind of like some of the work that we have been doing on the quality control of active translation, right? And most of my lab, again, studies the quality control of ribosome assembly and of active translation. And this is what we call, the bread and butter of our lab. This is what we, most of us study. And sometimes you have kind of a crazy idea. And when you have a crazy idea, very often you write on a piece of paper, you put it in your drawer, and you dream that one day you'll be able to kind of like explore this new scientific direction. However, I'm very fortunate that I'm part of Carnegie, where crazy ideas are absolutely encouraged, and where we have the intellectual freedom to actually pursue these like more exciting areas of biology. And so the kind of like last part of my talk, I'm going to tell you about this one of these absolutely crazy ideas that we've been working on. And this is, we're trying to tackle the question of whether all ribosomes are made the same. And you might have noticed that throughout my presentation, I have been drawing these ribosomes, these little cars, as being identical, right? That every single car that drives down these molecular highways are these little green cars. And this is because for the longest time, people believed that the ribosome composition is constant. However, there's very new exciting research suggesting that the ribosome composition can actually change, and there's subpopulation of ribosomes with different structure. So for example, these differences can be quite subtle. In this case, we have a car that's exactly like the green one, but now it's just painted pink. Or it can have a bumper sticker, or maybe there are new tires that someone put on them, right? Very slight differences. However, there are extreme scenarios where the entire ribosome composition changes. Right now, you have the sports car driving the highway together with these generic green cars. And this is like the extreme scenarios of ribosome heterogeneity that we're very passionate about in my lab. And although this heterogeneity has been observed in different organisms and in different tissues, the question that remains unanswered is, what is the function? Why would you have the cells build these different types of ribosomes, have these different kind of cars? And if you think about it, like, why do we drive different cars, right? Why do we choose to drive a Toyota Corolla versus, Tesla or a Hummer or something else? And there are a lot of factors that are involved, right? Maybe you live in different parts of the country, right? If you live in a big city, you choose one car. If you live in a more rural area, you choose a different car. Maybe your socioeconomic status affects what type of car you're driving. Or maybe your age, right? Because you wouldn't want to give your brand new convertible to your teenage daughter or something like that. So there's similar factors that can affect the ribosome choice for cells and organisms. Maybe organisms that undergo stress conditions such as starvation can assemble different types of ribosomes that help them overcome this environmental stress. Maybe the ribosome composition can also vary between different developmental stages. Like in certain stages of development, you choose to assemble a different type of ribosomes, or there's also the possibility of a tissue specificity. So these are all interesting research directions that we're pursuing, and we mostly study ribosome heterogeneity in a zebrafish. And zebrafish is absolutely fantastic model organism. There are many, many, many advantages to this tiny fish, but the two that are especially appealing to me are that this fish takes almost no time to develop, right? You can see here that in 2 days, you start from a fertilized egg to a tiny swimming larvae that you can observe. In addition, the laying the eggs, the fertilization, and then the entire development is external, and these fish are optically transparent. So you can use a microscope or your naked eye to see all these different stages of development. But what's even more exciting for us is that there's quite a remarkable ribosome heterogeneity in zebrafish. The very early stages of development are governed by these maternal ribosomes that are actually deposited by the mom. And then later stages of development in the entire adulthood of the fish uses somatic ribosomes for protein production. So again, these two types of ribosomes have been observed in zebrafish, but the functional consequences of having maternal versus somatic ones is very poorly understood. So we are devising very smart genetic tools and different fish lines to actually figure out why would you want to switch your ribosome composition and what is giving, for example, these early stages of development advantage for having a specific type of ribosomes versus the late stages. So, I usually say that I can talk your ears off about ribosomes, but I'll stop here, and I want to thank a lot of people who did really wonderful work for all of this project and continuing doing work in the lab. So this is a picture of the lab pre-pandemic. and we love talking about ribosomes, and we love eating cake. And this is a picture of us eating cake. And unfortunately, Zach and Hannah are not in this picture, and we couldn't take a new one because of COVID. But once we're back, we'll update it. But these are a wonderful group of people who make science really enjoyable and are really pushing forward all these 3 projects that I told you about today. I also want to thank Marty Halpern and Steve Farber in both Halpern and Farber Labs for teaching me everything I know about zebrafish. It's a very new model organism for me, and it's really fun working with this tiny fish. I want to thank a lot of collaborators at Carnegie, at Johns Hopkins, all over the country, and even our collaborators in Europe for contributing to different aspects of these projects, both for the ribosome heterogeneity and the ribosome collisions. I want to thank my funding sources. Everyone at Carnegie, it's a really wonderful place to work. And I want to thank my 1-month-old son, Daniel, and my husband, TJ, for helping me find time to put this presentation together and actually give this talk, because turns out that time is quite the luxury when you have a tiny baby. And I'd like to thank you for your attention and take any questions.
Thank you so much, Kamena. What a wonderful presentation, and you really made some complicated cellular dynamics very easy to follow. We have a lot of questions coming in, and to start off, we have a few questions related to everyone loves your car design metaphor, but some people want to know how you came up with it, and is the resemblance to a Volkswagen intentional or coincidental, and also people who are used to seeing the ribosome as 2 parts, does one car represent both of those parts in your metaphor?
Yes, absolutely. So the ribosome is composed of 2 parts, right? You have 2 subunits that come together, and they look like kind of like a macaroon, with like the French macaroon. See, like pastry analogies are much more near and dear to my heart versus the car. So I didn't even know it's a Volkswagen Beetle, but I will use this information once they update my talk. But yes, correct. The ribosome has 2 subunits, and they come together and kind of like clamp on the mRNA, or clamp on the highway. So the highway is actually in between them. And there's a very good car analogy having like these 2 subunits coming together. But this is the composition. And in this metaphor that I use, each car represents one functional ribosome that's composed of two subunits.
Thank you so much. And I love the macaron metaphor. Now I want to get custom macarons made for your lab. Next talk, when we can be in person. So we have a question from Amanda wondering, is ribosome function conserved in all life forms? Is this something that is in every kind of life that we see?
Yes, so the major function of the ribosome is to synthesize proteins, and it's very highly conserved. So everyone has ribosomes. There is no organism without a ribosome, right? But bacteria, archaea, zebrafish, humans, everyone uses ribosomes to produce proteins. So the difference is comes in this like kind of like what is the term? Extreme scenarios where, for example, you have different ribosome composition, and then the ribosomes can translate different messages in a different way. And one thing that's kind of interesting is that some ribosome components, for example, these wheels on the car, can now go and do other things in the cells, and they'll be different for different organisms. And we call this moonlighting function, right? Because their daytime job is to be part of the ribosome and make proteins, but when they have free time, they go and like bind other proteins or synthesize something else or serve as transcription factors. So they do something completely crazy. So that's why, and they'll be different between different species.
So the gig economy affects even cellular organelles. It's crazy.
Absolutely.
So we have a real back to basics question, but I think it could be useful for answering some of the subsequent questions from Joel, who wants to know the difference between proteins and amino acids.
Yes, so the amino acids are the building blocks of protein. So when you talk about a protein, you just have a very long sequence of amino acids glued together. And what the ribosome does is actually gluing them together, taking individual amino acids and putting them into long chains of proteins.
Thank you so much. I thought that would help clarify some things as we get into some of these more complicated questions. And then we have a couple questions from both William and Mike asking if there could be drug targets that target ribosome assembly or the ribosome function, and what kinds of diseases or pathologies might be targeted in such a way.
Yeah, I think all of you know about drugs that target the ribosome. Like 90% of the antibiotics that we take target the ribosome in bacteria. So the cool thing is that everyone has ribosomes, but for example, the ribosome in bacteria is very different from our ribosome. That's why when you take an antibiotic, your cells don't care, but the bacteria that are infecting you now cannot produce proteins and then they die. So this is a very huge class of molecules that we use, in the lab and also like in medicine for therapeutics. But they are very interesting, right? Inhibitors that specifically targets stages of ribosome assembly and also different steps of protein synthesis, also in higher organisms such as like our cells. And they're very, very useful for us as molecular biologists, but also hold like therapeutic potential. For example, like because protein synthesis is so essential, some of them have been used as, for example, cancer therapeutics, like such as cyclohexamide, which inhibits protein production. And because cancer cells divide, and they need a lot of proteins being made, by giving them cyclohexamide, you can actually hit them pretty hard. But it's a very non-specific type of approach, unfortunately.
And then we have two individuals, both Harold and Neil, are asking how maternal ribosomes are different, and if you could explain that in a little more detail.
Can give like an hour and a half talk on maternal and somatic ribosomes, but I'll try to synthesize it in like a shorter answer. So the ribosome is pretty cool, right? It's made of protein and RNA. So this molecular component, it has 4 RNAs and 80 proteins that come together in this big molecular machine. So the maternal and the somatic ribosomes differ in the RNA. So there's different RNA that is being synthesized and comes in the middle of the ribosome. And the cool thing about the ribosome is that the RNA is the business end. So if you think about it as the car analogy, the RNA is kind of like the engine of the ribosome. So these two types of ribosomes, the maternal and somatic ones, have different engines. So the questions we're asking, like, are they running faster? Are they more efficient? Are they better at protein synthesis? And also, are they cheaper to make, right? If you have kind of like a generic, I see like my car analogy needs to be, I need to do more reading to extend it, but some parameter of engine efficiency that you would look when you're buying a car, versus if you're me, you're going to look at what like the color is and other non-important factors. But this is like generally how they differ. And we did a very cool experiment with Nenad Ban at ETH in Switzerland, where we used cryo-EM to actually get the 3D structure of these two ribosomes. And they look so different. It's amazing because we talked ribosomes are so conserved that we usually use the structure of the yeast ribosome to build the human ribosomes, right? They're so conserved. And it's absolutely mind-blowing that in the same organism, you have two types of ribosomes that look so very different. It's really just a very extreme example of heterogeneity that we stumbled upon.
And so we have a question from Robert asking, in this quality control mechanism that you work on, using the car metaphor, if one highway is sort of stalled, are there parallel routes that can be used, or do you just have to wait for the highway to clear? Yeah,
so it's a great question. And usually you have multiple highways that would be encoding the same protein. So you can go to the other highways and use those to make the proteins, and then you just kind of like maybe even like close the highway and then degrade the entire thing. But there are extreme scenarios, for example, where that's the only highway you can use, right? That's the only piece of mRNA that's available to you. You need to translate. So there's very complicated mechanisms to actually resolve the accident, clear the accident, and then reuse the highway. So they're both possibilities in the cell.
Thank you so much. And then I have a question from Jeff asking, are the quality assurance mechanisms in place for ribosome assembly, are they more intensive, less intensive, pretty comparable to other kinds of quality assurance mechanisms in other cellular functions? Is this an area where the cell is extra cautious or just kind of average cautious to
Yeah. Well, we actually don't know that, right? Because ribosome quality control of ribosome assembly hasn't been really studied. The previous kind of like when we started this project, the idea in the field was that there is a final quality control step, which is called the test drive. And this is kind of like how this whole car metaphor evolved. But it's called the test drive because you build a ribosome and you export it in the cytoplasm, and then you take it for a test run. You make it drive a little bit, and if it's functional, you release it and it can go synthesize proteins. And if it's not, you put it in a trash can. And this kind of like made sense at the beginning, but then if you start thinking about it, that's absolutely crazy, right? Why would you put so much energy in building a whole car if step one, you make a mistake, right? Like why would you do that? So we thought maybe each step of this huge assembly line actually has a quality control factor that patrols it and like looks into that. And then because we make so many different mistakes, we engineer so many cells having different mistakes, it seems like that's the case. Or maybe they aren't like necessarily each individual step, but they're like very big central hubs where you make sure that what you are producing is forming properly before you proceed down the assembly line. But how it compares, for example, to the quality control in the endoplasmic reticulum or just general translation, we really don't know. But my gut feeling is that because it's so complex and has so many steps, I think it would be more complicated. There'll be more factors involved. But I'll keep you posted.
We look forward to hearing that you've answered that question in a few years. So we have a question from Alan who wants to know if viruses make use of ribosomes when they hijack a cell, if that is part of any viral tactics. Yes,
viruses are absolutely amazing. And just for like the non-viral connoisseurs in the audience, viruses actually, they don't encode their own ribosomes. So they don't have their own ribosomes, but they need ribosomes to make proteins. So when they infect the cells, they hijack the host ribosomes, and they're so efficient. So all the host ribosomes now abandon the molecular highways that are available to them, the endogenous highways that are available, and then go translate the viral proteins. And they have different tricks for that. Some of them use different messages. They encode like factors that kind of grab the ribosome and force it into translating their proteins. But there are as many strategies as there are viruses out there of how they can hijack the ribosomes and make them translate proteins.
Fascinating. I feel like viruses always yield the most interesting answers in these Q&As. They're always teaching us things, viruses. So we have two individuals who both asked more or less the exact same question of what is the lifespan of a virus? How many protein construction runs does a ribosome have in its life?
The lifetime of a ribosome.
Yeah, did I say virus? I'm sorry.
Yes.
My apologies.
No problem at all. So that is a really interesting question, and that's another kind of like crazy idea that we have in the lab of this of ribosome aging. So the ribosome is so the ribosomes in the cells are believed to be diluted and not degraded, which means that they live forever. So but there must be some ribosome turnover, right? You probably do not contain the ribosomes you're born with, unless you're very, very young. But there must be some turnover, but we really don't know, because the ribosomes are so abundant in the cells, and they're so essential that for decades we were not able to manipulate them. And it was really like technologies such as CRISPR, high throughput genetic screening, as gene editing, and mass spectrometry and cryo-EM that allowed us to now start looking into these questions, such as the ribosome aging. They probably age, they probably have a limited lifespan, they probably have a limited number of proteins that they can synthesize, but we don't know what that number is.
Everyone is asking such cutting edge questions that your lab is thinking about and demonstrating your creativity with. I love it. So we have a number of people, for probably unsurprisingly, are asking about something you just mentioned, which is CRISPR, since it just won the Nobel Prize last week. Very exciting. And people want to know how your lab uses CRISPR, and then more generally how CRISPR, as you just mentioned, how CRISPR has changed the work that cell biologists like you do.
Yeah, so we use CRISPR, we use actually a variation of CRISPR. So normally CRISPR is an enzyme, like Cas9 is an enzyme that serves as little scissors that can cut the DNA. And what my old lab did, what Professor Jonathan Weissman did, was break the scissors, right? So we use a version of CRISPR that doesn't cut the DNA. But it's actually, the enzyme is used as a scaffold to bring different activation or inhibition domains to different parts of the genome. So we can regulate gene expression. We can turn genes on, or we can turn them off. When we use this as a high throughput platform to screen for, in my case, factors that involve ribosome biogenesis. And CRISPR has really revolutionized biology. I'll give you one example. So CRISPR allows genome editing. In the case, for example, you can introduce a very precise mutation or a tag or something else in the gene that's encoded in the organism. So how did we do this before? Before, if you want, for example, to introduce a mutant, you put it on a plasmid or a lentivirus and put in the genome as an extra copy. So the downside of that is that this extra copy is so highly expressed that you have many copies of your mutants. For example, endogenously, your message is you have one molecular highway that's being translated by one ribosome, and now your mutant produces 100,000 highways that are translated by a ton of ribosomes. So your protein levels are so skewed, and it's so hard making conclusions, right? When you have such a confounding factor, and going very precisely into the genome and making these edits actually resolves this. So there are many other factors, and I actually feel so fortunate being a biologist right now, having these wonderful molecular tools that we can use. And sometimes with my lab, and we did this in my old lab, we read this like very old biology papers, and it's really, I'm in such awe with the scientists that came before us, because they had so little technology that they can take advantage of, and they made such big discoveries that it makes me feel bad that like I have all this technology. There's a lot of pressure that now we need to do. We do need to do better. We need to do more. But CRISPR is definitely like, one of the top things that allowed us to do the research in my lab, but I'm sure for many other labs.
I love that answer. That's wonderful to think about. The technology just keeps getting better and better, but when we look back at these foundational discoveries that were done with very rudimentary tools, it's really impressive. We shouldn't forget about it. So we have several people asking about if you could elaborate more on diseases that are caused by ribosome malfunction, particularly cancer. If you could talk a little bit more about how that works and the types of cancers that are most likely resulting from ribosome problems. Yeah, so there are a lot of diseases, like different types of cancer and different that neurodegeneration has been associated with
mutations either in the ribosome itself or some of the quality control factors. For example, the factors that we discovered that kind of like close the on-ramp have been mutated in various neurodegenerative disorders. So we're interested in how that exactly works. But for example, the most clear case of ribosome-associated diseases are the syndromes called ribosomopathies. They're quite interesting because usually in ribosomopathies, you have a defect in the ribosome. So you instead of you make half the ribosomes that you need. And the other half just get degraded by some unknown quality control pathway. And as we said, ribosomes are essential, protein production is essential, but surprisingly, people with ribosomopathies actually can live like a pretty long life. And early in life, they have anemias. And which makes sense, right? Because if you can't produce proteins, probably your red blood cells are the first ones to get hit because they actually make a lot of hemoglobin. So they synthesize. And if you don't have enough ribosomes, you wouldn't make enough hemoglobin and you become anemic. So this phenotype makes sense. However, as these people age, they start developing cancers, which is very surprising because cancer is a disease of cell division, right? So how are these cells dividing when they don't have enough ribosomes to make enough proteins? So this is a very puzzling question of how do you have a disease that starts as a hypoproliferation, like less division, to a disease that becomes a hyperproliferation, more division. So I feel like I answered your question with another question, but this is one of the kind of classic examples of ribosome related diseases. And we understand kind of like half of it, and there's a lot of work going into understanding how are these mutations in ribosomes driving the cancer. And one of the hypotheses is that now you're making ribosomes that are defective, so they don't translate very well, so they make mistakes. And the mistakes that they make in the proteins can facilitate the evolution of cancer, can facilitate the cancer initiation and progression.
That's interesting. So in that scenario, if the ribosomes were making mistakes, they wouldn't necessarily be making the same mistakes over and over. They could just be making all different mistakes. So that seems like it would be very difficult to study.
Yeah, and there's this really old paper about, and it's called like cellular catastrophe or something very dramatic, where it says that if you start making mistakes in the different stages of like the DNA, the RNA, the ribosome, it becomes a cellular catastrophe when the ribosome gets hit, because now you're just making so many mistakes so often that you just give the cell so many options of a cancer to evolve. Like eventually you will make the right mistake to fuel hyperproliferation.
It's like that old metaphor with the 1,000 monkeys on 1,000 typewriters. I'm sure you've heard that before. Eventually one of them will type Shakespeare. 1,000 faulty ribosomes, eventually one of them will make cancer.
Yes.
So we have a question referring to one of the answers of your previous questions, which is you talked about the material that ended up getting thrown in the trash during ribosome assembly. Is there a recycling process? Do those things get broken down and reincorporated either into another ribosome or into some other cellular machinery, or do they just get broken down entirely and thrown away?
Yeah, so it's an interesting question about this recycling, whether the cell is very good at recycling or if it trashes everything. So if the cell breaks the ribosome to its core components, the protein, which is, we're going to give amino acids, right? Because the amino acids build up protein, so you go the other way and just break it into amino acids. They can get recycled and get incorporated into other proteins. Same with the RNA, it can make nucleotides and they can be repurposed. But the more interesting question is that if you take a ribosome, because it has so many components, you can pull them apart, take the really broken ones and degrade those, but the ones that are salvageable, maybe you can like reincorporate into other ribosome. So these are questions. There's some evidence in the literature of this recycling process, but it's something that is definitely very interesting, and a lot of people are looking into it.
Thank you so much. So we have time for one last question for anybody listening in who we did not get to your question. You can email your unanswered question to events@carnegiescience.edu, and we will make sure those are forwarded to Kamena, and she will answer them for you, because we don't like to leave people hanging. And so we have a question about how stressful conditions could affect ribosome function. Is that a thing?
Yes. This is something that's also regulated very tightly, right? Protein production, because proteins are essential when you need them to survive, but also they're very expensive, right, to make. There's an estimate that about 50% of your energy goes into making proteins. So for example, if the cell is stressed, if you're starved, if you don't have enough nutrients, you don't want to be wasting energy making proteins, right? You want to be saving that. So cells do something very interesting. They relocate where the ribosomes are translating, what highways they're driving down. So they shut down translation on most mRNAs, and most highways get closed, and they drive only on the highways that can produce protein that cope with the particular stress factor. So there's this translation regulation that happens, and has been very extensively studied in bacteria and in yeast. But it's something that's true, like throughout evolution for our cells as well. But it's a very interesting topic. Great, thank you so much. And if you want to remember something from this talk, DNA is great. Ribosomes are much cooler. And thank you for giving me the opportunity to present our research and all the wonderful questions. Thanks.
Thank you so much, Kamena. That was a wonderful talk. Thanks for really providing, and you saw how interested the audience was with all those questions, and also congratulations. So thanks for being here. I'd like to ask everyone to mark their calendars, because our next event, our next talk will be a special event. Each year we co-host with the Kavli Foundation, the Royal Embassy of Norway, and the Norwegian Academy of Sciences and Letters, the Kavli Prize Lecture Laureate. And this year it's going to be Avina van Dushoek. She's a professor of molecular astrophysics at Leiden Observatory, and she's known for her work on the life cycle of interstellar clouds and the formation of stars and planets. And why this is so interesting is that she's set a lot of the groundwork for how we understand whether or whether not some of the exoplanets that we're now observing, thousands of them, may or may not contain the building blocks of life. So I hope you'll join us on October 28th, Wednesday at 4 o'clock. So we'll see you there. Thank you again, Kamena, for a wonderful, wonderful lecture, and we look forward to welcoming all of you back later in